All right, here we're going to review for the test. The test is Friday. I thought I'd go over some of the general things that are on the test. It is made, so I know exactly what's on it. So here is a picture of a gene. Now, these are the general parts that we'll be looking for. So of course, here's our structure. Now, the first thing I want to point out is that the exons are 5' and 3'. Now this is the reverse from, see here, this is DNA, double-stranded, and the transcription start site, the TATA box, all of that is going to be over here. And this is going to be what? 3' over here. But the UTRs are in the RNA. So this would be the 5' UTR and the 3' UTR. But up here, we are what? We are 3'. All right, and 5'. This is horrible to write with. Okay, so just keep that in mind, that the DNA is 3' to 5', but the UTRs, because they're in the RNA that will be copied essentially from this, are 5' and 3' in the opposite direction. Now, these are untranslated regions. These are regions that are before the start codon and after the stop codon. So before the start and after the stop. Now that's where they would be in the RNA. So the RNA is what? Is down here. This is the region that is copied. All right. This is the termination. This would be where we have our hairpin loop. All right. That would terminate transcription. But here is our translation stop. So this is where we might have one of those 3 stop codons, like the U, A would go in here, or UAG or UGA. But that's what this red line stands for. And here is our start codon, and that's where it's going to be a methionine, and it's what? An A, U, G as a start, but that's RNA. So what is the DNA right here? The DNA right here would be what? TAC in the actual DNA. All right, and over here this would be what? ATT in the DNA. So this is just to keep it tracked for you to understand what's going on here. Everything here that goes on in here, down at this level, well, maybe not this. Has to do with the DNA, but the rest of this is all RNA. So exons and introns and that sort of thing, that's done at the RNA level. You have to make the RNA before you can start splicing. And of course, the DNA is going to be up here where we have this indicating the double-stranded molecule here that it's kind of representing. All right, now what is in the DNA? Well, the transcription start site, the transcription factor binding sites, and the termination sites. These are all in the DNA. All right, DNA. These other things are more or less in the RNA. And now what kind of things are we going to have here? Well, the transcription start, this is where transcription is actually going to start copying. But way before that, or not way, but 30 maybe base pair before this, this is where you're going to get something like the promoter binding sequence. The promoter will be here, TATA. Something like TATA. All right, and here are your other transcription factors, maybe your GAGA and something else back in here. Maybe, and then way back here would be like an enhancer. Now the other thing that's not listed here that would be of interest to us are Cpg Islands, and the Cpg Islands can be into the first intron. And so all the way from here back, not too far back, but 500, what this would be about minus 500 in here, we can look for Cpg Islands. and that of course stands for a C followed by a G. This is a very rare combination. You never find them next to each other unless something interesting is going on in the genome. It's just they're not there. Because chemically, if you get a C next to a G, whenever there's transcription or something goes wrong, it gets repaired as the C turns into a T. So you almost never see these unless there's like a protein on it protecting it or something. So C's followed by G's, you'll find them in the promoter region, and maybe as many as 500 base pair back, but you can also find the Cpg island moving into the first intron. But that's in DNA. Remember, it's in DNA, and remember that the big deal is that the C's get methylated. So an open one is not methylated, but if I draw one and it's like this and colored in, okay, well, that's methylated. Okay, if it's left open, it's not methylated. If it's methylated, it's going to be sticky to the histone. All right, think of it that way. And therefore, this is going to stick more tightly to the histones. Therefore, it's going to block RNA polymerase from attaching to the TATA, and you are going to turn that gene off. If it's loose, if it's not methylated, it's on. The RNA polymerase can attach. So understand, here's where some of these pieces are. I just wanted to draw, here's like a little representation of it. The transcription start site, which not on the next test, but probably on the next, we'll get more into eukaryotic transcription start sites and how to find them. The real problem with them is most genes have more than one. So right now we're just talking about them having a single one, and in bacteria you're just going to have just one, but later on we're going to find that you have many in a eukaryotic gene, so it can be starting all over the place here, which really confuses the research when you get into it. You're going like, well, this RNA is not the same as the other one, and it's a problem. Anyway, so I just wanted to show you this here. Now, the other things that we will be covering is, let's see, we're going to cover, make sure you understand, let's see if I can, I think I have letters in this program. Let's see if I can erase this. Let's see here. Okay, that's easy. But I can type in this, I think. Yes. So the other things we need to look at are cell differentiation. All right. And development. They're the same thing, more or less. So stem cells. All right, let's do this again. Stem cells are only partially differentiated. Only some of the genes are unmethylated. Most of them are methylated and turned off. And as the cells age and environmental factors come into play, essentially they get unmethylated and turned on. And so you slowly, hormones, things like that, will turn them on. So when they turn these on from stem cells, we say that they differentiate. All right? And that differentiation when you're young is called development. So when you're it's a very generic term there. But later on, or as a general rule, you call this differentiation. When differentiation occurs as a normal plan of your growth or your development as an organism, so as you go from the womb to an adult, your cells are still undergoing development. So that's what we call that. But it's just a form of differentiation. Now, your cells can go through differentiation at other points in time. You get an injury, and the stem cells decide that they need to turn into a specific type to help you heal that injury. That's not development. That's just differentiation that's assisting you in a wound, in healing a wound. And we can induce differentiation. We can take stem cells out of your body, and we can convince them to become different types of cells. So keep that in mind. Now cancer, we need to talk about how this is related to cancer. All right, cancer. My reference would be when your cells are confused and you turn on genes at the same time that shouldn't be, or you turn off genes that shouldn't be turned off, and the cell doesn't die. The cell instead kind of goes crazy, and if whatever causes this causes two genes to be turned on at the same time, you essentially have a confused cell. Normally these cells will actually kill themselves, but there may be something that keeps them from doing that. I did tell you a story, I believe, about the smoking tobacco and benzopyrene, and or is it pyrobenzene? I never can remember. But the idea here is cancer. The story is the peanut butter and mayonnaise story. All right, that's essentially our explanation of cancer. Another thing that can lead to issues and can, well, I guess you would say it accelerates development or alters development, are hormones. Hormones are the actual chemicals, or one group of chemicals that can alter differentiation. So hormones, and the story there was the story about the little girl, and if dad was using the steroid, the testosterone stick, roll-on stick, like the deodorant, and the little girl were to have it touch her face, she would grow a beard, and that would be permanent. That's the we'll just call that the roll-on testosterone story. All right, and that's a story about how you need to be very careful about hormones, because especially in children, they're still undergoing development, and they are very sensitive to them, and you can differentiate the cells very easily, turning those cells into, essentially they behave like male cells in the face if they encounter certain types of testosterone, certain types of hormones. And the same goes with males and interacting with female hormones. So you have to be very careful with children with this sort of thing. All right. I did post a new video from Lucy Cantor, if you guys know her. Last year she made a little video on Grandma's Cookbook. So please watch that. You will need to know Grandma's Cookbook for the test. So listen, pay attention to her video. That's a really good way to kind of break it out, break it down on paper, know where, the ribosome translates. That's like what the cook reading the ingredients, that sort of thing. Make sure you can go back and forth between the cooking and the an actual translation and transcription, because that will be a question on the test. Let's see, what else do we need to know? We did the parts. You need to know, this is kind of separate here, know the difference between the old definition of a gene and the new definition. So, for instance, what we now know is that most of the differences between us, all right, most traits come from the promoter region, from differences, the promoter region, all right, and not, okay, I guess that's as much as it's going to let me type. Nope. All right, and not in the open reading frame, which is the part that translates to a protein. So you need to know that term, open reading frame. So in other words, it's not the part that says how to make the protein. It's not the list of ingredients. It's the it's when, how much, the timing, that sort of thing. What cells. Those are where most of the differences come, and that's all in, of course, the promoter region. So that's where most of our differences between us and between organisms come from, not from the proteins like we used to thought. So we pretty much need to redefine a gene. A gene is no longer what we thought it was, because a gene is said to be something that leads to different traits. And of course, the majority of traits don't come from what we have, or what we originally defined a gene as. So make sure you understand that idea. The other thing, the main idea, besides all of these things, you need to know these things, but you also need to know general transcription and translation. You also need to understand how to read the codon table. Besides that, you need the apartment and the mansion story. So on the website you have the apartment and the mansion, and so you need that story, and you also need, let's see, you're going to need the hippo, whale, or don't judge a book, don't judge. Let's see here. Don't judge story. Whoop. Dyslexic fingers. Alright, so don't judge story. You need the to make sure you understand the hippo and whale part, and the bacteria and archaea, and why they have been misclassified over the years, and why when we can sequence data we had to change what we were doing. It's not what you look like, but it's what your DNA says. Remember that. So you need to know those stories, the apartment, mansion, and the don't judge a book. Now, in relation to the apartment, mansion, you also need to make sure you understand that there are differences in how, or I should say where, maybe you would say, transcription and translation take place. So how the genes are organized are different, for sure. Of course, you don't have as many parts in a prokaryote, but the ribosome, okay, so make sure you know in prokaryotes, what? The ribosome goes to the RNA, and this is different from in eukaryotes. The RNA finds the ribosome, okay? And you can have multiple ribosomes on a prokaryotic mRNA, but there's only going to be one pairing, between 1-to-1 pairing, and so of course the result of this is that the prokaryote's very efficient, very fast. It can make proteins very quickly, it can divide very quickly, it does things quickly. Eukaryotes, of course, are slower. They can't do things as quickly, but they have more things, of course, that they can do. They have many more genes, more highly regulated, can do them at the correct time in the correct place. So there are advantages, all right? You do not have methylation as a general rule, at least not in the same sense as you do in eukaryotes. So prokaryotes are simpler, much less regulated. But in the next test, we will learn that they're still plenty regulated as we learn about operons. And that should do it. The only other thing, make sure you're aware of how to use dot plot. I will ask some questions on dot plot and BLAST. So know what a consensus sequence is. Let's see. Know what an alignment is. How you would make a consensus sequence out of an alignment of several genes. So if you know those things, you'll do fine on the test.